Swayam Prabha. Digital India. Educated India.
So welcome back everyone. We will continue our lecture on the internal mechanics of the microscope, and I'll also introduce the concept of fluorescence as well. Okay. Okay. So coming to the concept of fluorescence, all right. Have you heard this term before? Yes. What is it? Glow in the dark. What else? What else do you know? Okay, okay. So this glowing in the dark comes in many different mechanisms. There is essentially it's a transfer of energy. Okay, you put in energy in one form. You know, energy cannot be created or destroyed. It can only be transformed from one state to another state. Okay, so essentially a transformation of energy. Okay, so you put in energy in one form and it comes out in another form. Okay. So fluorescence is a type of energy transfer where you put in energy in the form of one type of frequency of light and it comes out as another type of frequency of light, another wavelength. Okay. So in nature you will have a lot of these examples. So for example, I don't know if you know, but all scorpions in the world, they are fluorescent. Okay. All of them. So if you happen to see a scorpion in the dark and if you glow a fluorescent torch on them, they will start glowing, the backs will start glowing. Okay, but it will not happen with the normal light, you have to glow with the fluorescent light. Okay. So that is what I am saying. Not everything is fluorescence. So there is other types of glowing feature which is called luminescence or phosphorescence. It all depends on the mechanism in which energy is transferred. For example, if in the night if you go to the beach, you will see the waves, and the waves as they are breaking on the shore, they will be glowing, right? But that is not fluorescence. There is phosphorus in the water of the oceans. And that phosphorus, because of that energy transfer, it glows, and that is called phosphorescence. Okay, and some of these marine animals that you see, they will glow in the dark. That is actually also not fluorescence. That is called luminescence. Okay, bioluminescence. Yeah, if it is from biological organism, it is called bioluminescence. Okay, so not everything is fluorescent. Fluorescence refers only to a specific type of luminescence where you shine light of a particular frequency, in this case UV, and then you get a light of another lower frequency. Okay, so only those type of glowing is called fluorescence. Okay, and so unless if you just shine a torch light on something and it glows, that is not fluorescence. Okay, so you have to know this distinction. So it occurs in nature in many forms. So animals which live in the very dark, in the very depths of the ocean, they often use this fluorescence as well to catch their prey or show signs to each other, all of that. Okay, and so one guy, he decided to investigate this and how we can use it for our biology. Okay, so there is a species of jellyfish. You can see this one here. And this guy, he was interested in what makes this jellyfish glow. Okay, what is there inside the body. So what he did was he collected a lot of these jellyfish from the ocean. Okay, and he then killed them and chopped them. Okay, and then he separated all the body tissues by centrifugation, and he tried to isolate all the proteins from that jellyfish. Okay, and he through chromatography and other techniques, he tried to separate all the proteins, and then he wanted to see which protein is responsible for this glowing feature. Okay, and then he discovered this particular protein called green fluorescent protein that is there in this organism. Okay, and then he started with a small number of jellyfish, but in order to get a significant amount of this protein, he needed to chop off hundreds of jellyfish like this, okay, in order to get even a few milligrams of this protein, okay. So, but you know, that is how people discovered this, okay. And then he did a lot of work as to why this protein glows, what is special about this protein compared to other proteins. You see this barrel-like structure. This barrel-like structure is somehow responsible for the transfer of energy that falls on this protein and the green light that it emits. Okay, so he did a lot of study on the structure of this protein, and he got the Nobel Prize for this. Okay, so that is how the world of fluorescence was actually. People knew that fluorescence is there, but what makes something fluorescent, that was discovered by this guy. Okay. Roger, same. Sorry? See, glow in the dark is a very broad term, and fluorescence is just a very specific type of glow. Yeah, technically that is bioluminescence, because the chemical reactions that are happening within the jellyfish's body, that causes it to emit this kind of signal. But if you isolate that particular protein like it did, and then if you shine UV light on this protein, then it will start glowing green. Okay. So this can be triggered both through enzymatic bioreactions or through shining of a light as well. That shining of a light is what is fluorescence. So if you if you see some deep sea scuba divers, and if they are carrying some light, and if they shine a light on some, let's say some marine organism or some reef or something, and if it starts glowing, then that is fluorescence. Okay. So as you know, this is our visible spectrum. Okay. So we are not going into this high frequency and low frequency ones. We are restricted only to a very narrow part of the visible spectrum, and within that visible spectrum we have light which is around 360 nanometer wavelength all the way up to about 750 nanometer wavelength. Okay. So all of these visible light spectrum that we see, we can utilize for fluorescence microscopy. Okay. Now, before we go into fluorescence plus microscopy, I want to tell you why do we need fluorescence and microscope? Why not only microscopy?
Better identify the sample. Fluorescence, and it's actually emitting light. Better to resolve the image and be able to see more clearly than if it wasn't fluorescent.
Yeah, you're partly correct. So under the previous examples of microscopy that I showed you, we were just using white light. Okay, it can be a light from a bulb, it can be a light from a candle, it can be a light from this kind of light, LED light. Okay, any light. These are all white light. Okay, so if we shine them, the light will pass through a sample, and you can get a picture in gray, black and white or gray. Or if you have some red color, you can stain it a red color or some other dye, you will get some color. The reason why we need fluorescence, why this guy is so important, he did not envision that it will be used for microscopy. He just wanted to know what is causing the jellyfish to glow. Okay, but what people after him did was that if we combine the concept of fluorescence with microscopy, then we can label things with specific colors. Okay, so for example, the first example that I gave you, that let's say you are observing the sky. Okay, there are clouds in the sky. Okay, the sky is blue in color, and then the clouds are generally white in color. Now let's say you see a cloud which is slightly gray or dark gray in color. What does that mean? Rain cloud, right? So because there is a difference in the color, now you are able to predict rain, right? If it was white in, if the rain cloud was also white in color, you would not be able to say whether it would rain or not, right? Because of the change in color, that is why you can make additional predictions of the nature of the cloud. So similarly, in a cell, our cell has many different things inside it. There is nucleus, there is mitochondria, there is the cell membrane itself, Golgi body, endoplasmic reticulum, so many things are there, right? So if all of them appear the same color, you will not know which one is which, okay? So we have to, if you are interested in knowing where is the mitochondria in the cytoplasm, where is the endoplasmic reticulum in the cytoplasm, are they close to each other, are they overlapping each other, are they far away from each other, are they interacting with each other? If you want to know this kind of question, then you need to put some additional labels on them, and that label comes from fluorescence, okay? So yes, that stain has to be yeah, so if that stain can stain them separately, you can, but most stains don't differentiate between them. So there are one example that I showed you earlier was that red color stain. So it will stain the nucleus in slightly dark purple and the background in light purple. That is the only difference you will see. But most of the stains that we see under light microscope, it's not possible to distinguish between the organelles of the cell. Okay. So for that we need to label it with fluorescence. Okay. So in fluorescence, what happens is each of these organs will have a
How are you able to actually label the different organelles with?
yeah, that's what I'm saying. So there is a principle called immunofluorescence. Okay, I'll just write it to you. So immunofluorescence comes from two words, immuno and fluorescence. Okay, immuno comes from a word which is I think you have heard of this, called immunology. Okay, and immunology involves the use of something called antibodies. Antibodies are a specific type of protein that can go and bind to a specific type of protein. Okay, so let's say I have a protein on the mitochondria. Okay, that protein is found only on the mitochondria and nowhere else, not on the nucleus, not on the ear, not on the Golgi body, nothing. Okay, so now if I know that this protein is there on the mitochondria, I will use an antibody that will go and bind to that protein of the mitochondria. Okay, and then I will that protein that I have, I will tag one fluorescent, let's say red fluorescent protein or green fluorescent protein to that protein. So then that protein is attached to so then that fluorophore. So let's say this is my mitochondria. Okay, so this mitochondria has a certain proteins on its surface. Okay. So now what I will do is I will take, let's say, green fluorescent protein, okay, and I will tag it to an antibody. This green fluorescent protein is the protein which is glowing, okay, and this antibody is so specific that it will go and bind only to the proteins that are there on the mitochondria. Okay, so now once this antibody goes and binds, then whenever I see GFP in my eye, I know that this GFP is bound to this antibody and this antibody is bound only to this protein of the mitochondria. So whenever I am seeing GFP, that means there it is mitochondria. Okay.
How does this help us overcome the resolution limitations of a normal microscope? Because we learned about Abbe's creation. And then usually when we talk about these things, we say that they're like smaller than wavelengths of light, right? So to resolve that, even though fluorescence microscopy will tell us that it is there, it doesn't tell us how many, it doesn't tell us anything about the dimensions or the shape of that.
Yeah.
So how do you overcome this?
Yeah. I'll come to that. Yeah. Yeah. So yeah, in the later slides, I'll come to how we get over that resolution. So the concept is clear, right? So if you want to study a certain part of the cell, of the organelle of the cell, we tag it to an antibody, and that antibody is tagged to a fluorescent protein, and wherever that protein shows up, we know that that is bound only to that organelle. Okay, so that whole concept is called immunofluorescence. Okay. So, depending upon what we want to see, whether it is the mitochondria or the cell membrane or the nucleus, we can choose a fluorophore like that and we can tag it like that. Okay. So, different parts of the cell will show up in different colors, and the range of colors available to us is wide. Okay. So, there are so many colors in this range. So, each organelle can be labeled with different colors, and you can see it under the microscope in that color. Okay, so just to give you a brief introduction about the mechanism of fluorescence, how it works. Essentially, this molecule, which is called a fluorophore. Okay, it will come up in the next slide. So it is called a fluorophore. Fluorophore is a molecule that is able to absorb light of a certain wavelength. Okay, and its electrons, which are in the ground state, they will get excited to a higher energy state. Okay, and then they will hold that energy only for a few nanoseconds, and then they will fall back to their ground state. Okay, as they are falling back to their ground state, that loss of energy, that energy that they are releasing, that comes out in the nature of fluorescent light. Okay, when the electrons rise up to this excited state level, there is some loss of light due to some atomic effects, and the remaining light, so let's say I shine blue light on this molecule. Okay, so it will rise up to this level, it will lose some energy in the excited state, but then from the lower excited state it will fall down to this ground level, and because there was some loss of energy, that blue light that I shine comes out as green light here. Okay, you know that blue light has a little more energy than green light, right? So if I shine green light here, then it will come out as orange light. Okay, if I shine orange light, it will come out as red light. Okay, so a little bit of energy loss happens. Because of that, the wavelength of the light shifts slightly. Okay, and that is called the Stokes shift. Okay, so I shine light of a particular frequency, and the Stokes shift happens because of some energy loss, and then within a few nanoseconds you will have the fluorescence coming out of that molecule. Okay, and that this formula describes it, the number of photons that are emitted here, okay, divided by the number of photons absorbed here, that is called the quantum yield. Okay, so when people so the GFP that is isolated from that jellyfish, that will have a certain quantum yield, but that may not be good enough for our real world application. Our real world application, we may need fluorophores which much higher quantum yield. Okay, so people do a lot of chemistry and lot of designing of chemistry to achieve materials of higher quantum yield, so that even with a few photons of light that you give on that, you will get a huge amount of photons that are released. Okay, and so if you see the whole spectrum, there are lot of fluorophores which are available. So there is a tryptophan. Tryptophan is an amino acid that is found in many proteins. So if you look at any molecule, any protein molecule in itself, if it has tryptophan in it, it will also glow in another color. Then there are colors which are in the blue range, UV range, the blue range, the yellow, orange, red, all of these. So depending upon how many colors you want to use together, you can choose, pick and choose each of them. Okay, so let's say I have a cell. Okay, let's say I have a cell like this. This is the cell membrane, and here is my nucleus. Okay, and let's say I want to image the mitochondria. The mitochondria is like distributed all all around it. Okay. So now if I want to see this under the microscope, what will I do? I first need to label the cell membrane with a particular fluorophore, so that I can see, okay, this is the boundary of the cell. Okay. Then I need another fluorophore, another color, to label the nucleus. Okay. So that it will label the DNA in the nucleus, and that will appear another color. Okay. And then I need a third fluorophore, third color, to label all the mitochondria. Okay. So using this, I can identify which is the boundary of the cell, that is the cell membrane, that is one color, nucleus is another color, and mitochondria is another color. Okay. So from this list, this is a small list, but from this list of fluorophores, I can choose which colors I can use for which part of the cell. Okay. Is this clear so far? Okay. So now I am just giving you the some examples of some fluorophores. So what you are seeing here, let's say DAPI. Okay. DAPI is this chemical structure, and DAPI is generally used for staining the nucleus. Okay. So this nucleus will usually appear blue. Okay. So whenever you are seeing any fluorescence images and you will see blue nucleus, you know that typically it has been stained with DAPI. Okay. If you need green color, fluorescein is a very good, very common example for green color, and then Texas red is a very common example for red color fluorescence. Okay. And then there are some other examples as well. Tritici is also red in color, acridine orange, you know, orange color. Propidium iodide is also red in color. Okay. So different molecules are there which have different colors, and people use them in combination with each other. So that is a chemistry question. So I don't know the answer to it because I am not that much. But whenever some photons are falling on these molecules, they their vibrational states they have their vibrational states in such a way that these molecules transfer their energy. So let's say a photon falls on a particular molecule. Okay. Depending upon their vibrational states, the energy gets transferred to the other molecules. Okay. And there is a temporarily reconfiguration of the molecular arrangement of these molecules here, but only for a few nanoseconds. Yes, yes. So how I showed it here before, here the photon is falling on the electron of one of the atoms in that compound. Okay. And the electron is jumping to a higher energy state and then losing some energy and falling down and emitting that particular state. Now whether it will be responsive in the blue range or green range or orange or red, that depends on the structure of the compound itself. So different compounds react to different things. For example, DAPI. DAPI is, you can see this purple line is the DAPI. Okay. So if you shine light of around 360 nanometer on DAPI, then DAPI gets excited. Okay. And then it releases energy in the 400 nanometer range. Okay. But if you shine some green light on red light on DAPI, it will not get excited. It will get excited only in this range. Okay. Around 360 nanometer. Yeah.
So here PRIDC, it has blue, red and yellow fluorophores.
No. That is just the different atoms. That is not the color. It's just to differentiate between them. Okay. Yeah. So similarly, Tritici is this green one. You have to shine light of 488 nanometer on Tritici. Only then it will get excited, and then it will emit light in the around 420 or 430 nanometer range. Okay. And Tritici is again around 560 or 580 nanometer, and then it will emit light in around 600, 620 nanometer. Okay. So people often use this blue, green, red combination very well, so that you can shine light of specific wavelength and you can get light of specific wavelength. Okay. So this separation also needs to be there. Okay. So if you try to have two blue or purple dye close to each other, then it will be difficult. Okay. So let's say you say I want nucleus to be blue, but I also want the mitochondria to be purple. That will be difficult because the emission signal of both of them will overlap with each other, and you will the same light can affect both of them. Okay. So for example, if I say that I want nucleus in this DAPI, okay, so you shine this DAPI, but there may be another dye which is also very similar to DAPI, okay, so it will overlap with DAPI and it will simultaneously excite both of them, so you cannot distinguish it easily. Okay, so it is necessary to have some degree of separation, blue, green, red, or blue, green, orange, or green, yellow, red, something like that. Okay, so you have to choose your dye accordingly. Okay. Okay, so these are some examples that I am showing you. So first thing is you can see the nucleolus. Nucleolus is a part of the nucleus. So it is stained with a blue fluorescent protein. Okay, so BFP is blue fluorescent protein, and then the mitochondria is labeled with cyan fluorescent protein. Okay, it's a slight variation of the blue, light blue. Okay, and then vimentin is another protein in the cell that is labeled with green fluorescent protein, and then the Golgi body is stained with also another green, yellow fluorescent protein, but it is labeled in a pseudo color. Then the endoplasmic reticulum is labeled in the red fluorescent protein, and the nucleus is labeled in the red. Okay. So where the nucleus is labeled, you can also see the nucleolus in the nucleus. So those are the different examples. Okay. Now if you want to stain multiple colors, you are seeing this cell here. Okay. The nucleus is stained with the cyan, the something in the cell cytoplasm is stained with green, and something is stained with red. Okay. So wherever it is overlapping, you can see that those objects are there. Okay. Same thing here, the nucleus is in yellow, I think this is the ER, and then this cyan is another color and the ER is in red, or maybe the mitochondria is in red and the cyan is some other color. Okay. And then for example here you can see the nucleus is red in color, and these are actin filaments. These are like the cytoskeleton, the skeletal fibers that are there within the cell, so they are labeled in green. Okay. So you can see these different organelles within your cell. Okay. And using this you can get different information, like which organelle is close to which organelle interacts with which organelle. Is the mitochondria close to the nucleus or is the mitochondria far away? Okay. If the cell is having a lot of metabolism, its mitochondria will appear in one shape. If the cell is not having a lot of metabolism, its mitochondria will appear in another shape. So using this kind of information, you can identify what is the state of the cell, whether it's a healthy cell, whether it is behaving abnormally, whether it is producing its proteins properly, whether it is doing its metabolism properly. All of these things you can identify directly by looking through your eye. Okay. Okay. So any questions up till this point? All right. So now we go into the structure of the microscope itself. Okay. So typically a microscope will look like this. Okay. On the outside you will see like this, but if you make a cross section, you will see the way the light is passing through. So in this case, this central part is the stage. Okay. This is where you keep your sample. It can be a glass slide, it can be a well plate, it can be a petri dish. Okay. You keep it on the stage here. This stage can move in the X and Y or even in the Z direction. Okay. This part is called the binocular. This is where we look through our eye and this part. Okay. This part is the binocular. This is where we have the eyepiece. Okay. This part is called the condenser. Okay. And this is the light source. This light source shines the light. It passes through a series of filters, turns by 90 degree, and then goes down through the condenser and falls on your sample. okay, and then your light passes through the sample into this thing, okay. This thing is called the objective, okay. This objective is the most important thing in the microscope because this one has all the convex lens inside it, okay. So this is the heart of the microscope, and that light from your sample passes through the objective magnifies it, and then it either sends it to your eye or it can send it to the camera that is attached to the microscope, okay. So you can take a picture, alright. So this kind of diagram is called the light path that follows inside the microscope. Okay. So if you take a cross section of the objective, you will see something like this. Okay. Somebody was asking about the corrections. So if you take an objective like this, you will see this kind of information. The company, these are called Nikon. Okay. So there are 4 major microscope companies in the world. You can note them down. So Nikon, Olympus, Zeiss, and Leica. Okay, so these are the 4 major microscope companies in the world. Just like how you have Google, Facebook, Apple, and all. These are the 4 big microscope companies. They make almost like 99% of all the microscopes in the world. Okay, these 4 together. So, huh? What? Yeah. Huh. So electron microscope is a different field. I am talking about light microscopes. Okay. So Zeiss was started by a guy called Carl Zeiss. He was a German guy. He used to make a lot of these lenses, and then he started this company Zeiss. So these 2 are German companies. Okay, and then these 2 are Japanese companies. Okay, so these Japanese companies started with making cameras and films, and then they gradually got into the business of making microscopes. Okay, but these are the original 2 microscope manufacturers, and they started in Germany because they are very old companies, and these are 2 relatively new companies which started it, but they also make very good microscopes. Okay. So anyway, whenever you buy a microscope, you will need to buy this kind of objectives. Depending upon what kind of sample you want to see. If you want to see human cells, then maybe you need 20x, 40x, or 60x. If you are seeing bacteria, then you definitely need 100x. Okay. So according to that, you have to buy these kind of objectives, and so they will come with the name of the brand, and then they will come with the magnification. This plan apo is a kind of label that tells you what kind of corrections are there in the objective, whether there is spherical correction or chromatic correction. Those that comes in, that is the designation for that. Okay. Some objectives will have this kind of label called 1.40 oil. Okay. So sometimes what happens, you have to add a drop of oil on this lens here, so that the light, as it passes from the sample, it gets bent through the oil. The oil has a special refractive index, so it passes through the oil, and then it passes through the series of lenses here. Okay. So it will be mentioned whether you need that oil. Okay. And then some more information, it will be written as DIC, with which kind of light images it can produce. So sometimes it will be written pH, so phase contrast. Sometimes it will be written DIC, so differential interference contrast. And then you will be seeing something called WD, that is the working distance. So working distance of 0.21. That means this objective has to be within 0.21 millimeter of your sample in order to get your image. Okay. So you have to know all of these details. Each of these objectives cost anywhere between 3 lakhs to 10 lakhs. Okay. So you have to know all of these details very well before you buy your microscope. Okay. You can't just buy. So whenever you are buying a laptop or some computer, you go through all the specifications, right? how much storage, how much all these things. So how much RAM and all that. So similarly, when buying a microscope, we have to know all these technical details, and then we buy the microscope. So if you take a typical objective and you do a cross section, you will see a multiple lens inside it. So as someone said, a correction, aberration. So all of these lenses are placed subsequent to each other, and the correction, the aberration happening in one is corrected or compensated by the lens that is behind it. Okay, so this light, as it passes through, it gets diffracted and again recombined as it passes through the lens. Okay, so that is how these microscopes are made. So the people who make these lenses are very specialized, and they are there only in these two countries. Okay, so all of the lenses that get made, they need very nanometer scale workmanship. Okay, so it is those people are very sought after, very much in demand, and they are very well paid. And so all the microscope manufacturers in Germany and Japan, they depend on those people to grind these lenses to that very special perfection. Okay. Okay. So here is I am talking about a little bit about the correction. So two types of correction are needed. One is the color correction, and one is the shape, the curvature correction. Okay. So as the light is passing through, the colors get split up. So let's say I am seeing the blue nucleus and the green mitochondria. Okay. So if I don't correct the colors, then if I see through the eye, the blue will look here and the green will look here. Okay, because the light of different colors are bent to different extent. So a certain correction is needed so that both of them appear in the same plane. Okay, so that is what is called chromatic correction. So that is what is done here. Okay. So chromatic aberration, so different microscope objectives, some can correct for 2 colors, some can correct for 3 colors. Okay, and then there is spherical aberration. So sometimes a flat object can appear rounded. Okay, so when you look under a binocular or some cheap binocular, you will see, if you look at it, you will see things are bulging in the corners. It is curved. So that is because of this spherical aberration, because different parts of the lens are bending light to different extents. So in order to compensate for that, again there are different lenses. So depending upon how much correction I want, I can buy objectives of that particular scale. Okay, so each of them will be more and more expensive, but they will be better in terms of the correction. Okay. The colors can also get bent.
Colors can get bent, sir.
Yes. So if this nucleus, let's say I know that this nucleus is circle in shape. Okay, and it is labeled with the blue color. Okay, so now this nucleus can appear like this if I don't do any correction. It can get slightly rotated. Okay, so that color correction is needed so that the blue and the green appear on the same plane, and the spherical aberration is needed so that the nucleus appears round shape instead of the distorted shape.
Then what's field curvature, sir?
Huh? Field curvature is this entire field. If this is your total view, then things that appear on the corner, that should also appear straight. So that is what is meant.
Yes.
Okay. So in a microscope, we also need some kind of filter. Okay. So when we are seeing light of different colors, we are not seeing them together. Okay. So let's say I have 3 colors in my sample. Okay. I first want to see the blue color. Okay. So what I'll do is I'll first shine UV light. That UV light will go and shine on the blue fluorophore, and it will give me the blue light. Okay. So at that time, I don't want to see the green, I don't want to see the red, I will only see the blue. Okay. Now I take a image or capture an image of the blue color. Okay. and then I move on to the green. Okay, I captured that green, and then I move on to the red. I capture that red. So I take three different things, three different colors. I take one at a time, and then I recombine them together to get this kind of fluorescent image. Okay, so all of these colors are taken separately. They are not taken at the same time. So this color is taken first, this is second, and then all of them are combined together to give you one composite image. Okay, so in order to do that, when I am shining blue light, I don't want any other light to be there. So I need filters to block out all the other lights. Okay, so that is what is done in these kind of filters. Okay, so for each color I need one kind of filter that will block out everything else and only allow that particular light to be observed at a time. Okay, so depending upon which dyes we use, we need to buy these different kinds of filters. And then the last thing is this light source. Okay, so earlier people used to, when there was no bulb and other thing, people used to use candle light or some other light. Then gradually bulb was discovered, electric bulb, and then these fluorescent lamps light were introduced. And then much later on people started using mercury or metal halide lamp, and then even later lasers were discovered. Okay, so now people use different kinds of lasers, and the advantage of laser is that lasers come in specific frequency. Okay, so this light that you are seeing, this has many different kinds of frequencies. Okay, but lasers come in one particular frequency. So, and lasers have much more penetration in them. Okay, and so with the laser light you can image much better. And let's say I have blue color, green color, and red color in my sample. So for blue color I will use this laser, for green color I will use this laser, for red color I will use this laser. Okay, so with shining one color laser I will capture one type of color in my sample. Okay. Okay, so I am not going into this too much. This is about the camera. So cameras come in different forms. So once you study about semiconductors, you will know that generally there are two types of camera. There is SCMOS camera, and then there is EMCCD camera. Once you know a little bit about semiconductors, you will get to know. But essentially these cameras have very high sensitivity. The main job of the camera is to convert the photons into electrons. Okay, so the photons that are coming from the sample, okay, we need to capture them in the camera. So these camera that you see, all these cameras have semiconductors in them which convert the photons of the light into the electrons, and that electron is what you see on the screen of your computer or your LCD display or LED display. Okay, so that is what the job of the camera is. And the better the camera, the better it can convert the photons into electrons, the better is the camera. Okay, so all your iPhones and all, they have good cameras. Yeah.
So here you're only talking about CCD image sensors, but isn't it over the past decade, all CCD sensors have been replaced by CMOS sensors?
Yes, that's correct. Yes.
So in the lab, do you still use CCDs or do you use CMOS?
In certain application, CCDs are important, especially when you have to capture fast moving objects. Then CCD is better than CMOS. Yeah. So let's say I want to capture the video of a bacteria that is swimming very quickly. So in that case, I want to capture the motion of its tail or the cilia. So if I use a CMOS, I cannot get that rapid frame rate. I will miss out on all the frames in between. So I use a CCD to capture the duration of the tail of the bacteria.
Correct me if I'm wrong, but isn't it like because CCD sensors they take snapshots, all the crocodiles take snapshots at the same time, whereas the CMOS they are sequential.
Correct. Yes. Yeah. Okay, so this is all about the standard setup of the microscope. You need a light source, you need objective, you need your sample stage. The light will go from the light source, pass through your objective, and then get magnified in the objective, and then it will either come to your eye or it will go to the camera where you can take the picture. Okay, so that is the basic principle of all microscopes. Okay, so now we'll come to some advanced type of microscopes, and one of them is called confocal microscope. So confocal microscope comes in because let's say I want this, let's say my object is shaped like this, okay, a thick object, and let's say as another example I have, I'm just borrowing this, let's say I have two objects, okay, I want to image this and I want to image this, okay. So now if I put this under the microscope, let's say I can capture this whole bacteria, rod shaped bacteria in the microscope in one plane. But let's say my bacteria is like this. The microscope objective has a certain working distance, it cannot go above and beyond, and this is a very thick sample. So how will I image this under the microscope? That is very difficult. So what a confocal microscope does is, it takes a sample like this and it captures the fluorescence, it excites the whole object in one go, okay. But the emission that happens, the emission is filtered out, okay. So it will take the emission, there is a small pinhole in a confocal microscope. All the emission from this goes through that pinhole, and that pinhole filters out this region and this region, and only a thin section of this goes through a confocal microscope, okay. So you can actually see the section of a thick object in a confocal microscope. So let's say another example, let's say I have a loaf of bread, okay. In that loaf of bread there are 10 slices, okay. All of them are packed, stacked on top of each other and packed in a plastic bag, okay. Now I want to see the 5th or 6th slice only. I am not interested in the top and not interested in the bottom. Let's say there is some, in the 5th or 6th slice, there is some fungus growing. I want to see it. What will I do? I will try to take the whole, I will try to make my microscope in such a way that I will take the picture of the whole slice, but then I have the ability to go slice by slice, okay. So I will take the first slice, second slice, third slice, and when I come to the 5th or 6th slice, there I will see the fungus is growing in the bread, okay. And then I will know that is the whole section, okay. And then I can reconstruct all that and create a 3D view of the whole loaf of bread, okay. So that is the concept of confocal microscope, okay. And so all the things that I was talking to you about is called wide field. Now we are going into confocal. So in a confocal, whatever I am showing you is, let's say this is a pollen grain, okay. This is a pollen grain, and I am taking slice by slice image of the pollen grain, okay. And there are 12 slices, and these 12 slices can be recombined, and you can see that this has this kind of spike features on the surface of the pollen grain, okay. If you look at it under a regular microscope, it will look like this. You will not be able to see that much details, okay. But if you look under a confocal microscope, you can see the internal structure and also the outside structure, all the spikiness that is there on the outside, okay. So that is the advantage of a confocal microscope.
Sir, how does it know that the inside is green in the confocal microscope?
How does it know the inside is green?
Yeah, so did you like slice it or something, or?
So that is the thing. So we have not sliced it, but we are, when the microscope is taking the image, the camera is capturing the image, it is capturing only one slice at a time. Okay.
So it captured that without slicing?
Yes, without physically slicing. So that is called optical sectioning. How do we know that?
No, but how does it know that it's green and that it has to be
So the color is coming from the fluorophore, okay. So the inside is labeled with one color and the surface is labeled with the another color, okay. It is shining the light through the whole thing, okay. So the light that is falling on the whole thing, but when it is capturing the image, it is taking light only from one plane at a time. Okay. because that light is passing through a pinhole, a very, very small hole, and that hole blocks the light from the top and the bottom plane. So it will take only one plane at a time. So using this, we can reconstruct this kind of image. Sorry. There is another kind of microscope that is called a 2-photon microscope. In a regular microscope, this is your objective, okay, and this is your sample, okay. Your light is coming and falling and exciting all the fluorophores that are there in your thick sample, okay. But in a 2-photon microscope, you will have two separate photons that go and strike your sample at only one point, okay. And because the possibility of two photons simultaneously hitting one electron is very, very rare, so the amount that you excite is very, very small, okay. So maybe you can see this is the very small point that is fluorescent, okay. Okay, so you can image, if your sample is this thick, you can guide this spot of light one at a time, and you can image one at a time all of this, okay. So you can reconstruct this at a time, okay. And this is useful for imaging a deep tissue, okay. So this is a section of a brain of a mouse, okay, and all these green objects are the neurons in the brain, okay. And you can see the thickness of your sample is about 2 millimeter, okay. So you can see some of the neurons which are on the surface, they are of one shape. Some of the neurons which are in the center, they are of another shape. Then there is another set of neurons which are of different orientation, and then another set of neurons which are of another orientation, okay. So different slices of the brain have different types of neurons, okay. So you can resolve them in 2-photon microscope, okay. And then there is another example which is total internal reflection, okay. So I told you about that in the previous thing. So you can use that same principle in microscopy as well. So in total internal reflection, what happens is you have light or laser coming in, and instead of passing through your sample, it gets totally reflected back, okay. So your light is coming through your objective, falling on your specimen, and again going back into your objective, okay, totally internally reflected. And with that, you can see, and there is something called evanescent wave that forms, and that evanescent wave is used for imaging things which are at the very, very thin surface, like a few nanometers of your sample, okay. So in a regular wide field, you will see some features, but under total internal reflection, you can see some other features which are not as prominent as in the wide field, okay. So you have to, with this total internal reflection, you can see additional things in your sample, okay. And then there is something called light sheet microscopy, okay. So instead of light as a point, you are making light as a sheet, okay. So how you do this is that you have two objectives which are facing each other, okay, and the light coming from one is passing through the other, okay. So light forms like a thin sheet, and that thin sheet of light passes through your whole sample at a time, okay. And with that, so instead of, you have a thin sheet of light, and if this is a sample, it is passing through the whole thickness at a time, and you can get the whole image of your sample in one single shot, okay. And this is the kind of image that you see. So this is a whole zebrafish, okay. This is a fish about this much in length, okay. And if I use a traditional microscope, it will take a long time to view all of these, capture all of these images, okay. But if I have a sheet of light that is passing, in one quick, in a few minutes, I can capture the whole thickness of the light, okay. And so, yeah, so when I have very, very large organ or organisms that I want to image, I can use the light sheet microscope to image this kind of objects. So this is a comparison of all the different techniques. So let's say your object is like this. If it is a wide field, you are seeing the whole thing. If it is a confocal, you are having a point source of light that is scanning and capturing the image. if you are having a 2 photon, you are exciting only this point and you are scanning it. And if you are having a total internal reflection, then you can see only this part of your sample, only the bottommost part. And if you are having a light sheet, then this whole sheet of light can scan from the top to the bottom quickly, and you can get the whole 3D view of the object. Okay. So these are the different microscopy techniques that we use in our biology. Okay. Questions? I know this was a lot of technical information, but if you want, I will share these slides with you. You can search up some of these technical terms, and so yeah, microscopy in general is really beautiful, because with your own eyes you actually get to see what is happening at that. And if you combine it with the concept of the colors and the fluorescence, then you can get so much information that it will fascinate you, and it will ask you, you will really be able to ask a lot of biological questions, why things happen in the micro scale. Okay. Photo bleaching is, if you continuously shine light on a fluorophore, after a certain time its sensitivity gets reduced, it will get bleached, because there is some permanent chemical reaction that happens when you shine light on a fluorophore, and because of that there is irreversible damage that happens on the fluorophore. So if you take a fluorescence sample and you image it after 10 times, 20 times, you will see that its fluorescence getting decreased over time.
Sir, can you say that confocal microscopy causes more photo bleaching than two photon?
Correct, yes, it's correct. Because if you see this, image, the confocal microscopy is shining this whole thing, right? Let's say I want to image only this part, okay? So, but the confocal microscopy is illuminating this part and this part unnecessarily, which is a waste. So, if I use a 2 photon, I can only illuminate this part, and these 2 parts are unaffected by that. Hope you learned some new things today. Thanks.
Swayam Prabha. Digital India. Educated India.